Hello everyone, I am Saurabh and I welcome you back to the Digital Board series by Orchids Biosciences. This is the third video for Heredity and Evolution. If you haven't watched the previous videos yet, I have provided its link in the description below, so please do look for it and watch it. In this video, we are going to look into the following topics. We will start with dihybrid cross and then learn about Mendels Laws of Inheritance. Then we will discuss about sex determination, in which we will discuss about the sex determination in human beings in detail. So let's begin with the dihybrid cross. So what is a dihybrid cross? Di means two. So a dihybrid cross is such a cross which involves the analysis of two independent traits or characteristics. So cross which involves the study or analysis of two independent traits is called as a dihybrid cross. These two independent traits can be any two independent traits. For example, we have studied 7 pairs of contrasting traits. So we can take any two pairs. For example, we can take as the color of seed and shape of the seed. So for example, these two characteristics or these two traits we are going to study, color of the seed and shape of the seed. So this cross which is involving two characters or two traits will be called as a dihybrid cross. What was a monohybrid cross? Monohybrid cross involve any one character. For example, if we take only the color of the seed, then it is a monohybrid cross. But here we are taking two independent traits. One is color of seed and one is shape of seed. So not necessarily we need to take these two traits only. We can take any two traits. So we will call it as a dihybrid cross because here two independent traits will be observed. So let us perform a cross and see between color of seed and shape of seed. So what are the two alleles for color of seeds? So for color of seed, the two alleles are yellow and green. Whereas for the shape of the seed, we have two alleles. They are round and wrinkled. So these are the two alleles or the alternative form of the same gene. So one allele is round, the another one is wrinkled. Similarly, here one is yellow, another one is green. So we can see two pairs of contrasting characters which we will be studying here. Among round and wrinkled, we know round is dominant and wrinkled is recessive. Similarly, yellow is the dominant character and green is the recessive one. So this is the basic information which we already know. We will be using this information in the dihybrid cross. So let us begin a dihybrid cross in which one parent will be dominant, that is round yellow, and another parent will be recessive, that is wrinkled green. So let us start with the parent generation. We know that in parent generation, one of the parent will be purely dominant, which means it will be having the pure dominant characters, whereas another parent will be having pure recessive characters. So if we take one of the parent plant, the shape of the seed will be round, which is the dominant character, and the color of the seed will be yellow. So the character of the seed will be round shaped seeds and the color of the seed will be yellow colored seeds. So this will be one of the parent. There will be another recessive character parent also, that will be wrinkled, that is the shape of the seed, whereas the color of the seed will be green. So this is the plant with the dominant character seed, whereas this is the plant with the recessive character seed. And we have to be very sure that we take only pure line plants. We should not take any hybrid, which means the round will be capital R, capital R. So we have to take a plant which is pure line. So round is the dominant character, yellow is also the dominant character, whereas wrinkled is the recessive character and green is the recessive character. So for wrinkled, we will be representing it as small r, small r. Why not W? Because round and wrinkled belong to the same gene. So for the same gene, we have to use the same letter. So wrinkled, we will be representing it as small r, and for green, we will be taking small y, small y. So if we see, the genotype for the parent generation will be capital R, capital R, capital Y, which will be crossed with small r, small r, small y, small y. Or we can say round yellow seed plants will be crossed with wrinkled green seed plants. So this is the parent generation. Next, before going into F1 generation, let us look at the possibility of gametes. In monohybrid cross, we saw that each gamete will be receiving only one allele. But here if we see in dihybrid cross, there are two genes or there are two characters. This is one and this is another one. So from each of the gene, one allele will be going into the gamete. So among these two alleles, one allele will go to the gamete, and among these two alleles, one allele will be going to the gamete. So in dihybrid cross, when we are forming the gametes, we will be writing two alleles, one from this one and one from this one. So let us write what are the possibility of gametes. So we can see that maybe this allele will be present and this allele will be present, which means these two will be in combination. So it will be capital R, capital Y. Or else there is a possibility that this allele is present along with this allele. So again there is a possibility capital R, capital Y. Now what are the other possibilities? So we saw that with the first allele of the first gene, there is a possibility of being with the first allele of the second gene or also with the second allele of the second gene. Similarly, now let us look at the second allele of the first gene. So this can be with this one as well as this can be with this one. So it will be capital R, capital Y, capital R, capital Y. So in all the cases, if we see, the condition is going to be same. Why? Because it is a homozygous condition parent. So if we see, for all the gametes, the condition will be same because it is a homozygous parent. Next, if we look into the wrinkle green condition, here also two genes are present or two characters are present. So the first allele of the first gene can go with the first allele of the second gene, or the first allele of the first gene can go with the second allele of the second gene. So it can be small r, small y, small r, small y. So these are the two possibilities first. Next, there is another allele in the first gene. So this can go with the first allele of the second gene, or this can also go with the second allele of the second gene. So again, small r, small y, small r, small y. So these are the possibilities of gametes. So if we see all here, it will be same, capital R, capital Y. Here also all will be same, small r, small y. Why? Because the parent is in homozygous condition. Now if we cross this with this or this, the answer will be same. Or if we cross this one with this or this, the answer will be same because all here are same gametes and here also same gametes. Actually there are 16 possibilities which will come because here 4 are there, here 4 are there. Each can be combining with any 4. So 4 4s are 16 possibilities are there, but the answer will be same because here also gametes are same, here also gametes are same. So we will be writing only for one and we'll keep it as into 16. So if we see the F1 generation, we will be getting RY plus RY. So this will be giving us the F1 generation. That will be capital R, small r, capital Y, small y. This will be 16 times. Because here we have 4, here we have 4. So if we make Punnett square for this, we will be getting 4 into 4, 16. So 16 this we will be getting. So if we see in the F1 generation, our genotype will be capital R, small r, capital Y, small y. If we write the phenotype, we know that capital R is dominant over small r. So we have to write round because capital R is round. Next, capital Y is dominant over small y. So we will be writing yellow because capital Y is yellow. So round yellow will be the character which will be seen in the F1 generation. So this is about the F1 generation. Now what will happen when we self cross or perform selfing of the F1 generation? What is selfing? Selfing means crossing two plants belonging to the same generation. So if we take F1 generation two plants and cross them, we'll be calling it as F1 selfing. So what happens in F1 selfing? We'll be crossing a F1 plant with another F1 plant. So let us see what will be the possible gametes now. Same how we produce the gametes here, for this also we will be producing the gametes. So let us see what are the gametes possibility in F1 generation. We know these two belong to the same gene and these two belong to the same gene. Similarly here also these two belong to the same gene and these two belong to the same gene. So one allele will be coming from this one and one allele will be coming from this one. So let us look at what will be the possibility of gametes. First case, maybe the first allele of this gene may go with first allele of this gene or also can go with the second allele of this gene. So it can be capital R, capital Y or capital R, small y. So this is the first situation. Next, the second allele of this gene can go with the first allele of this gene or it can also go with the second allele of this gene. So small r, capital Y, small r, small y. So these are the possible gametes. Here also if we do, we will get the same because the genotype is same. Still, we will do it once and see for better clarity. This one can go with this as well as this. So it will be capital R, capital Y and capital R, small y. Next, the second allele. This can be with this and this can be also with this. So small r, capital Y, small r, small y. So these are the possible gametes. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. And it will be similar for the next parent also. Why? Because both the parents which we are taking are of the same genotype. So the gametes produced by them also will be similar. So these are the 4 possibilities. So here 4, here 4. Again, if we cross 4 into 4, we will be getting 16 combinations now. So we will perform the cross and see, because here there are different gametes. So we need to put up Punnett square and see. Here they were same gametes. So we knew that the answer is going to be same. So we directly wrote it without putting the Punnett square. But here we can see there are different gametes. So we'll put a Punnett square and see what happens when we cross one F1 generation with another F1 generation in a dihybrid cross. So we will quickly put all these gametes in the Punnett square. First we will put the gametes of one parent in these 4 boxes, then for the next parent in these 4 boxes. So let us put all the gametes now. They are capital R, capital Y, capital R, small r, capital Y and small r, small y. So these were the gametes of one of the parents. Similarly for the other parent also same gametes. So we have written all the gametes. Now let us look at the possibilities. So what if this gamete combines with this gamete? Then the resultant progeny will be capital R, capital R, capital Y, capital Y. What if this gamete fuses with this gamete? Then it will be capital R, capital R, capital Y, small y. Next, what if this gamete fuses with this gamete? It will be capital R, small r, capital Y, capital Y. Why didn't I write small r, capital R? Because we are following the denotation of first writing the dominant trait, then writing the recessive trait. Next, what if this one fuses with this one? It will be capital R, capital Y, small y. Again, dominant recessive. In that format we are writing. Next, what if this one fuses with this one? So it will be capital R, capital Y, small y. Similarly, we will be fusing all the gametes and writing the combination. It will be here capital R, small y, small y. Here it will be capital R, capital Y, small y. Here it will be capital R, small y, small y. Here it will be capital R, capital Y, capital Y. Here it will be capital R, capital Y, small y. Then small r, capital Y. Next, small r, capital Y, small y. Next, capital R, capital Y, small y. Next, capital R, small y, small y. Next, small r, capital Y, small y. Next, small r, small y, small y. So these are the possible combination of the gametes which we will get. So here what we are doing, we are crossing one F1 plant with another F1 plant to get the F2 generation possibilities. So these are the various possible genotypes for the F2 generation which we received now. Now let us look at the phenotype also. Here capital R, capital Y, capital Y. So here it is dominant character completely. Here also it is dominant character completely. So dominant is round and yellow. Moving on to the next one. Capital R. So both are dominant alleles. So the resultant also will be dominant. So it will be round. Capital Y, small y. Among capital Y, small y, capital Y is dominant and the dominant character will be expressed. So yellow color of the seed will be expressed. Next, capital R, small r. Here one is dominant, one is recessive. Among them the dominant one will be expressed out. So therefore round shape of the seeds will be there. And again capital Y, both are dominant. So obviously the resultant also will be dominant. So it will be a yellow colored seed. So round yellow. Next, capital R, small r, which means here dominant allele is also there, recessive also is there. Again the dominant allele will be expressed. So here also round and capital Y, small y. Again capital Y is there. So therefore it will be yellow. So we have got 4 round yellow so far. Let us look what are the other combinations. Capital R. So definitely it is going to be round because there is no possibility of having a recessive character. Next, capital Y, small y. One dominant allele is there, that's why it will be yellow. Capital R. So therefore it will be round. Small y, here there is no dominant allele, only recessive allele. And when both recessive alleles are present, it will be definitely recessive. So instead of yellow now, we will be having green color of the seeds. Next one, capital R is dominant, so definitely it will be round. Again, capital Y, small y. Capital Y is dominant, so it will be yellow. Next, capital R, small r. We know capital R is dominant, so that character will be expressed round. Now here if you see again small y, small y, which means it will not be yellow, it will be green because both are recessive alleles. Next, capital R, small r. Capital R is definitely going to be round. Capital Y, it is going to be yellow. So round yellow. Next, capital R, small r. Capital Y, small y. So it will be round because capital R is present, and yellow because one capital Y is present. Even if one allele is dominant, the phenotype will be dominant character. Next, small r, small r. Now if you see small r, no dominant allele is present. So what we will be putting here, we will be putting wrinkled character, not round, because both are smaller. Next one, capital Y, therefore it will be yellow because both are dominant. Next, small r, small r in the sense wrinkled, both are recessive. Next, capital Y, small y. Since capital Y is there, it will be yellow. Moving on to the next one. Capital R, small r. Capital R is there, that's why it will be round. Capital Y, small y. Capital Y is there, that's why it will be yellow. Next, capital R, small r. Capital R is there, that's why round. Small y, both are recessive, so therefore it will be green. Next, small r, therefore it will be wrinkled because not even one dominant allele is there. Capital Y, small y. Capital Y is there, that's why it will be yellow. Last one, small r, both are recessive, that's why it will be wrinkled. Again, if you see the next character also, small y, both are recessive, that's why it will be green. So now we got all the possible phenotypes for the F2 generation. What are the possible phenotypes? It can be round yellow, it can be round green, next it can be wrinkled yellow, and one more possibility we have, that is wrinkled green. So these are the 4 possible phenotypes which we can get. Now let us look at how many possibilities of round yellow. Round yellow 1, round yellow 2, round yellow 3, round yellow 4, round yellow 5, round yellow 6, round yellow 7, round yellow 8, round yellow 9. So if we see, there are 9 chances of getting round yellow out of 16. So 9 round yellow seeds. Next, round green. How many round green? 1 round green, 2 round green, and 3 round green. So 3 round green seeds possibility is there. Next, how many wrinkled yellow? Wrinkled yellow, wrinkled yellow, wrinkled yellow. So 3 wrinkled yellow and only 1 wrinkled green. So this is the ratio, the phenotypic ratio of the F2 generation, that is round yellow is to round green is to wrinkled yellow is to wrinkled green. So the phenotypic ratio of the F2 generation in a dihybrid cross is 9 is to 3 is to 3 is to 1. So this ratio is very important. Just like how the ratios were important in monohybrid cross, similarly the ratio of the F2 generation in dihybrid cross is also important. So this is about the phenotypic ratio of the dihybrid cross for the F2 generation, that is 9 is to 3 is to 3 is to 1. We have done the cross, performed it, and understood how the ratio is also coming. So this is about the dihybrid cross. So far we have seen both monohybrid as well as dihybrid cross. For the monohybrid cross, if you haven't seen that, please go and watch that video. In the previous video, I have explained clearly about the monohybrid cross, and in this video we learned about the dihybrid cross. Moving on to the next topic for this video, that is the Mendels Laws of Inheritance. Under the Mendels Laws of Inheritance, we'll be studying 3 laws. They are, first one, that is law of dominance. Second one is law of segregation. And the third and the last one is law of Independent Assortment. So these are the three laws of inheritance by Mendel which we will be studying. Actually, while explaining these laws, Mendel was using the term factor, because that time the word gene was not discovered and people didn't know what was something that was passing on from the parent generation to the next generation. So while explaining all these laws, Mendel used the word factor and explained. But for our understanding, we will be replacing the word factor with genes and alleles so that it is clear to us. But we have to remember that when Mendel explained all these laws, he used the word factor and explained the law, because he didn't know there's something called as gene which was being passed on from the parent to the offspring. He knew there is something which was being passed, and this was called as factor by him. But we will not be using this word because we know the words clearly, they are genes, alleles and all. So using the words which we know, we will be understanding the Mendel's laws of inheritance so that it is easy for us to understand the concept. The law of dominance and law of segregation can be explained by the Mendel's monohybrid cross, whereas the law of independent assortment can be explained by Mendel's dihybrid cross. We have already discussed Mendel's monohybrid as well as dihybrid cross. So again, with the similar or the same example, we will take and understand these laws. So let us begin with law of dominance. What is this law of dominance? This law states that in a heterozygous condition, what is the meaning of heterozygous condition? In a hybrid condition, for example, capital T, this is not a heterozygous condition, whereas capital T, small t, this is a heterozygous condition. So this law states that in a heterozygous condition, the allele whose characters are expressed, we know whose characters are expressed, that is capital T's character is expressed, that is the tallness is expressed out. We have already discussed this in monohybrid cross. So the allele whose characters are expressed, this is the one whose characters will be expressed. So the allele whose characters are expressed over the other allele is called as a dominant allele. So capital T is which allele? The dominant allele. And the characters of the dominant allele, this character which is expressed, this expressed character is called as the dominant character or dominant trait. Whereas the allele which is not expressed in the presence of another dominant allele is called as a recessive allele, and this character is called as the recessive character. And this is only the law of dominance. Law of dominance states that in a heterozygous condition, the allele whose character is expressed over the other allele is called as a dominant allele, and this character is called as the dominant character, whereas the other allele whose character is not expressed in the presence of another dominant allele is called as the recessive allele, and this character is called as the recessive character, and always the dominant character will be expressed phenotypically. So this is the law of dominance. Moving on to the next one, that is law of segregation. What is this law of segregation? This also we can understand with the monohybrid cross. So we will take a monohybrid cross, tall plant crossed with short plant. Tall plant is capital T, short plant is small t, small t. What are the gametes possibilities? Capital T, here also small t, small t. So this is about the parent generation. This is the parent generation's gamete. Then in the F1 generation, what do we get? We get only capital T, small t. Now when we cross F1 generation with another F1 generation, what do we get? Capital T, small t crossed with capital T, small t. So now when we see here gametes, that is of a hybrid plant, the gametes will be pure. That is, here will be pure capital T. Here there is another possibility of getting pure small t. Here also similarly, pure capital T, only one allele will go. Here also pure small t, only one allele will go. So if we look at the gametes, they are only pure. They are always pure with only one of the allele from the parent generation. So out of these two alleles of this gene, only one allele will be going to the next generation. So here there is no mixing. If there is any kind of mixing in this generation, there will not be any kind of pure T which will be coming for the gametes. So there is no mixing of characters in a hybrid, and the gametes which will be formed will be pure gametes. This is why it is also called as law of purity of gametes. Why purity of gametes? Because the gametes here are very pure. There is no mixing of character. If in case there was any mixing of character, then this will not be having a pure T, or this will not be having a pure small t. So what is this law exactly about? So this law is exactly about the fact that when two traits come together in a hybrid pair, this is one trait, this is another trait. So there are two traits which are coming in a hybrid pair. These two traits will not be mixing. So there is no mixing of characters in a hybrid pair. And when this hybrid forms the gametes, each gamete will receive only one of the allele. Either it can receive capital T or it can receive small t, which means there has been no mixing. It is very pure. The gamete which is being formed is pure capital T or pure small t. There is no mixing. So there is no mixing of characters. So this is called as law of segregation, or also called as law of purity of gamete. Why it is called law of purity of gamete? Because the gametes which are formed will be very pure. It will be purely capital T or purely small t. So this is the second law. Moving on to the last law, that is law of independent assortment. So what is this law? To understand this law, let us go back to the dihybrid cross which we discussed. For example, we can take any two characters, but we will be using the same character which we studied so that the explanation is easier. Let us look at the F1 generation. Let us directly start from the F1 generation. We knew that in F1 generation, the plants were capital R, capital Y, small y. Then what we did F1 selfing, that is crossing two plants of the same generation. What we did, capital R, capital Y, small y, crossed with capital R, capital Y, small y. So then after this we got the gametes, right? So this is what we did in the dihybrid cross. So this I am explaining with the help of a dihybrid cross, where the characters I have taken is shape of the seed and the color of the seed. Now what is the possibility of having a gamete? This allele of this gene can go with this allele of this gene, or can also go with or assort with this allele of this gene. So this allele of this gene's assortment with any of the allele of this gene is independent of this character, which means there are two characters, shape of seed and color of seed. So this is the first character which we have taken, this is the second character which we have taken. So this gene's alleles assortment, or assortment means simply going with, we can say, to pair with, we can also say. So this gene's any allele can go with any allele of this gene. It is not dependent on this, it is independent. I will make it more clearer. For example, we know this is a dominant character, that is capital R, that is round. This dominant character can assort with the dominant character of another gene, or can also assort or go with the recessive character of another gene. So whether dominant is present or recessive is present doesn't matter. Irrespective of that, independent of the second gene, this can go and assort with any of its allele. That is the meaning. Which means there can be capital R, capital Y also, as well as capital R and small y. Which means the assortment of one allele of a gene with any of the allele of the other gene is completely independent of what that gene is, which means these two genes are sort independent of each other. This is a dominant character, so it can either go with dominant or recessive, irrespective of the fact it is dominant or recessive. Now if we take capital R, small r, capital Y itself, so this allele can go and assort with this allele, or this allele can go and assort with this allele, or else this allele can also go and assort with this allele, or this allele can also go with this allele. So we can see this gene's alleles assortment is independent of this gene's alleles, which means if here it is a dominant character, that is capital R, irrespective of the fact this is dominant, recessive, both dominant, it doesn't matter at all. It can go and assort with it or go with it independently of the second gene. So if there are two genes which are present, this is one gene, this is another gene, alleles of two different genes get sorted into the gametes. So we are seeing they are getting sorted, or they are going with each other to come into the gametes. So the alleles of two different genes get sorted into the gametes independent of one another. They are not dependent on each other. So this is the law of independent assortment. So they are assorting, but it is completely independent of the second gene or the other gene which is present. With this, we are coming to the end of laws of inheritance. So moving on to the last topic for heredity, that is sex determination. So how is the sex of a newborn individual determined? For example, we can see the environment as a deciding factor for sex determination in case of turtles. Here, the temperature at which the fertilized eggs are kept will determine the sex of the newborn individual. If the temperature at which the fertilized eggs are kept is cold, so at cooler temperatures, the egg which will hatch out will give rise to an individual that is male. Whereas, if the fertilized eggs are kept in a warmer conditions, then the female will be hatched out. So here the temperature at which the fertilized eggs are kept will determine the sex of the turtle offspring. So this is how environment plays an important role in sex determination. So this is one of the examples for environmental sex determination. While in some animals like snails, the sex is neither determined by environment nor it is genetically determined. The snails can change their sex. Therefore, the sex is not genetically determined here. The sex can be changed from male to female or female to male. So we have seen a condition where environment decides the sex, a condition where it is neither environmentally determined nor genetically determined. Now let us discuss about the condition where the sex of the child is largely genetically determined, that is in case of human beings. So in case of human beings, the sex is largely genetically determined. To understand sex determination in human beings, we need to know some basic concepts. So let us learn about the basics first, then we will go into the cross. In human beings, there are totally 46 chromosomes, out of which 23 chromosomes come from our father and the other 23 comes from our mother. We can call it as 46 chromosomes or can also call it as 23 pairs of chromosome. Out of 46, 23 from father and 23 from mother. Now, out of this 46 chromosome, 44 chromosome or I can say 22 pairs will be same in case of both males and females. And the same set of chromosomes are called as autosomes, which are same in case of males and females. Whereas there will be the remaining two chromosome or we can say one pair of chromosome which will be different. And this one pair of chromosome which will be different in case of males and females are called as allosomes or also called as sex chromosomes. So allosomes are the chromosomes in males and females which are different, whereas autosomes are the chromosomes which are same in case of males and females. So autosomes are same chromosomes whereas allosomes are different chromosomes. There are 22 pairs of autosomes whereas only one pair of allosome or sex chromosome. So this one pair can be different in case of male and in female. In male they will be X and Y, whereas in females they will be X and X. So in female if we see they are same, whereas in male they will be different, X and Y. So this is the basics for the sex determination in human beings. So we will see the sex determination cross in human beings by taking a father and mother parent. So first we will take a father, we will cross it with a mother, which means basically a male parent is being crossed with a female parent. So as I already said, the autosomes will be same. So 44 autosomes for father plus the sex chromosome. What is the sex chromosome in male? They are X and Y. For mother, 44 autosomes will be same, just like father. The sex chromosome will be different. They will be X and X. Now, what are the gamete possibilities in this? 44 autosome, gamete will be having only half of it. So, 22 autosomes will come plus out of X and Y, only one will come, X. Next, the remaining 22 autosome plus the another one, that is Y. So, these are the possibility of gametes for male. For female, what are the possibilities? 22 autosome plus one of the X, the remaining 22 autosome plus another X. So if you see, in male only the Y chromosome is there, in female the Y chromosome is not there. So this male's Y chromosome or male's X chromosome will only determine the sex of the child. Why? Because in mother it is same, both are X and X. the male's X fuses or the male's Y fuses will only determine the sex of the child. So if this one fuses with this one, it will be 44 autosome plus XX. Now if this one fuses with this one, it will be 44 autosome plus XX. Now this one fuses with this one, 44 autosome plus XY. What if this one fuses with this one, 44 autosome plus XY? So if you see here, for female it is same, X and X. The male's X is going and fertilizing or the Y is going and fertilizing will only determine the sex of the child. So let us see how the child will look now. 44 autosome plus XX. XX means female, so this will be a girl child. Next, 44 autosome plus XX. XX means female, so this will be a girl child. 44 autosome plus XY, so this will be a male child. 44 autosome plus XY, so this will be a male child. So whenever the Y chromosome containing gamete goes and fuses with the egg, it will be a male child. If the X chromosome containing gamete goes and fertilizes the egg, it will be a female child. So if X goes and fertilizes the egg, then the child will be a female, because female egg will already be having the X, and again one X from the male is going and fertilizing, so XX female will be formed. Now what if Y goes and fertilizes the egg? Already the egg will be X, now Y is going and fertilizing, so XY male will be formed, because female will be having the XX sex chromosome, whereas males will be having XY sex chromosome. So if you see, there is a 50-50% chance. So if you see the ratio here, male is to female or female is to male, there are 2 males and 2 females, so the ratio will be 1:1. So there is a 50-50% chance for having a male child as well as female child. It is depending upon the father or the sperm, which sperm will go and fertilize the egg. If the sperm is having 44 autosome plus X, then it will be a female. If the sperm is having 44 autosome plus Y, it will be a male, because females are always XX. They do not have that Y chromosome to determine the sex of the child, whereas the males are XY, which means if X goes and fuses with the egg, it will be female. If Y goes and fuses with the egg, it will be male. So the sex of the child is determined by the male parent. So this is about the sex determination in human beings. Now let us quickly recall what we have seen in this video. We have started with a dihybrid cross, a cross which involves the study or analysis of 2 characters. Here we studied the 2 characters as the color of the seed, shape of the seed. Then we discussed about the cross, where first we took capital R, capital R, capital Y, capital Y. We crossed it with small r, small y, small y. Then we got the F1 generation, capital R, capital Y, small y. Then finally in the F2 generation, we got the ratio of 9:3:3:1. After the dihybrid cross, we discussed the Mendels Laws of Inheritance. In Mendels Laws of Inheritance, we studied 3 laws, out of which 2 can be easily explained by the monohybrid cross. They are law of dominance and law of segregation. And the law which can be explained with the dihybrid cross is law of independent assortment. So these are the 3 laws which we have seen. After this, we discussed about sex determination, where we first saw how environment can play an important role in the sex determination of an individual, where we saw the example of turtles. If the turtles' fertilized eggs are kept in a cooler condition, then it will hatch a male turtle, whereas if it is kept in a warmer condition, then the turtle which will be hatching out will be a female. So the temperature at which the fertilized egg is kept will determine the sex of the newborn individual. After that, we also saw that in snail the sex is not genetically determined. They can change between male and female sexes. And finally, we saw the sex determination in human beings, where the sex of the individual is genetically determined, where there is a 50% chance of having a male child and 50% chance of having a female child. It all depends upon the male parent, because males have 44 autosome plus XY, whereas females have 44 autosome plus XX. It is same in case of female. So whether the X goes and fuses or the Y goes and fuses will determine the sex of the child. So the sex of the child will depend upon the male parent, not the female parent. And the sex of the individual in human beings is genetically determined. With this, we come to the end of this video as well as heredity. Thank you so much for watching this video. If you like this video, please hit the like button, share it with your friends, and do subscribe to our YouTube channel.